Hi there. I'm so glad that you've tuned in. I'm going to read to you an article that is going to explain so much. It's from phys.org, 3rd of April 2019. It's called Metabolically Engineered Organisms Could Sustainably Produce Ingredients for Natural Foods, Flavors, and Fragrances. Always sustainably. Metabolically engineered organisms could sustainably produce ingredients for natural foods, flavors, and fragrances. Is that floral scented natural shampoo from the health food store good for the planet? Demand for organic food and fragrance is growing, but harvesting the ingredients used in natural products is putting a strain on limited environmental resources. One solution is to turn to microscopic organisms that produce natural ingredients in vats of low value fermented feedstock. Microbes produce nature identical compounds, while synthetics are a mixture of isomers, which have a similar structure to naturally occurring compounds. They aren't quite the same, explains A-star microbiologist Nick Lindley. In addition, a few tweaks to the metabolisms of the right microscopic organisms can power industrial level production that often outperforms chemical synthesis or extraction in terms of production rates and cost effectiveness. Sharp output increases from yeast and bacteria are already being recorded at the Biotransformation Innovation Platform, or Biotrans, the part of A-Star's food, nutrition, and consumer care cluster focused on compounds for the health, seasonings, and personal care sectors. Lindley moved to Singapore from France 2 years ago to lead the platform. Since its launch in 2016, Biotrans has focused on 5 goals for the future of food and fragrance, says Lindley. These aims are to maximize product yields through its microbial metabolism platform, accelerate product development to get products to market faster via its fermentation and downstream platform, discover new compounds, biosynthetic pathways, and microbial strains by trawling A-Star's vast natural product library, advance the development of healthier sweet, savory, and other flavorings as part of the Biotrans taste receptor platform, investigate flavors, fragrances, and metabolites by combining mass spectrometry with organoleptics, the study of sensory perception, to strengthen its analytical platform. The sweet smell of success. Improvements in microbe production rates have steadily emerged from the Biotrans microbial metabolism platform. In 2017, after increasing the output of a lycopene overproducing E. coli strain, researchers reported unprecedented yields of 2 high-demand natural aroma compounds, alpha ion 1, known for its sweet violet-like aroma, and beta ion 1, which produces a scent often associated with violet or raspberry. On the upper end, Biotrans researchers beat a yeast-proven beta ion 1 production rate, improving output 80-fold using E. coli. The 2 ion ones are used in everything from perfumes and oils to ice cream and maraschino cherries. Lycopene, the carotenoid pigment that makes tomatoes red, is one of the main precursors of these types of apocarotenoids. Carotenoids have global market worth that is expected to reach 1.53 billion US dollars by 2021. The complex metabolic characteristics of apocarotenoids have been a challenge to produce in the laboratory. In their quest to optimize output, the Biotrans team divided E. coli's apocarotenoid producing biosynthetic pathway into 4 sections or modules and optimized enzymatic steps in each through genetic and enzyme engineering. The group described their method as a plug-and-play system, meaning that the platform is capable of producing various apocarotenoids simply by changing 1 or 2 genes without perturbing the whole strains, says lead author Simon Kungquang Zhang, a Biotrans research fellow who supervises several translational research projects. This system has been shown to also work on a number of similar substances, including lycopene, beta-carotene, phytoene, and retinol, vitamin A, an ingredient that will be familiar to those who use wrinkle creams or acne treatments. Patenting the system is the next step, and Zhang reports that there are, quote, quite a few companies in discussions with us to co-develop the bioprocess for commercialization, end quote. Meanwhile, Biotrans scientists are also examining whether product range development can be pursued internally with a view to spinning out on their own commercial venture. Accelerated antioxidants. In 2018, Zhang led another study building on his apocarotenoid work. The group used E. coli to produce a 1,600% improvement on current methods for producing the antioxidant astaxanthin, another type of carotenoid, and the red pigment that makes salmon and shrimp pink. Astaxanthin exhibits strong antioxidant, anti-inflammatory, and anti-cancer activity, and Zhang points out that its biological activities and benefits are supported by more than 50 human clinical trials and more than 1,000 papers published in leading journals. As a result, it is widely used in food, feed, nutraceuticals, cosmetics, and medicine. The global astaxanthin market is predicted to reach $2.57 billion by 2025. Most of the astaxanthin produced commercially is derived from algae such as Haematococcus pluvialis. This freshwater unicellular green microalgae transforms into red cysts under stress conditions, including nutrient deficiency, salinity, and high temperatures, in combination with high irradiance. But the algae is slow growing and usually produced in closed clear tubes, photobioreactors, to reduce cross-contamination from microorganisms such as microalgae, fungal parasites, and zooplankton predators. As a result, producing astaxanthin requires a relatively large amount of land and costs more than $7,000 per kilogram. By building on the same modular approach used for apocarotenoids, the team from Biotrans and the National University of Singapore developed a complex self-learning system known as a multidimensional heuristic process, an MHP, capable of balancing the enzymatic activity within and among different modules simultaneously. Using this approach, in Nature Communications, the team reported a Nature-identical astaxanthin production rate that was 16 times higher than the algae-based production rate, with an enantiomeric excess, a measure of purity, of 100%. The main advantage of the MHP is it enables the systematic but rapid optimization of biological systems to achieve high yield production of metabolites, such as astaxanthin, Zhang explains. This system is now on the cusp of commercialization. Our current technology is able to produce astaxanthin at high concentrations and low cost on a small scale, says Zhang. The next challenge is to scale up our process in large fermenters with a capacity of up to 10 cubic meters and purify the compounds effectively. Zhang also points out that the team's MHP has been used to optimize many different metabolic pathways and molecules. Used with mathematical models, it could improve the performance of many microbes, he says. It has already been applied to produce of nerolidol, well known for its fresh, woody scent and frequently used in shampoos, body lotions, and soaps, and linalool, a compound with citrus tones used in many cleansing lotions, aftershaves, and hair care products. With the MHP E. coli produced linalool at 680 times the rate of a previously engineered yeast strain. Searching a trove of tastes. Biotrans researchers are also on the hunt for new food ingredients. Over at the Biotrans Taste Receptor platform, senior research fellow Ann Coy and her colleagues mine A-Star's rich resources for molecules that refine taste sensations, work that might provide people with more options so that they can make healthier choices. To find these molecules, Coy trawls A-Star's Natural Product Library, NPL, one of the world's largest resources for the study of biotechnologically useful microorganisms, genes, enzymes, and other bioactive compounds. Established in 2013, the NPL houses more than 37,500 plant samples, 123,000 microbial strains, and 270,000 extracts derived from these specimens collected from more than 100 countries. According to researchers at A-Star's Bioinformatics Institute who manage the NPL, the collection represents, quote, 57% of all known cultured fungal genera, 67% of the world's plant families, and 70% of the filamentous bacterial genera, end quote. Genome sequence data for more than 150 microbial strains at NPL have already been uncovered, with potentially thousands more in the pipeline, and plans are underway to develop a digital database that will enable researchers to probe microbial genomes in ever greater detail. To make use of this huge resource, Biotrans researchers have developed cell systems that mimic human taste responses to sweet and bitter tastes, enabling researchers to quickly evaluate thousands of molecules for their taste modulating properties. Cell systems are faster and cheaper than human sensory panels, groups of people who give feedback on taste and sensation. By further improving these high-throughput screening approaches, Coy hopes to accelerate discoveries of novel molecules, adding to the pipeline of molecules that are safe, healthy, and effective for use in the food industry. Of particular interest are molecules to help address increasing rates of diabetes. Half of Singapore's population will have type 2 diabetes by 2050, according to a multi-institutional study published in 2014. To combat this disease, an increasing number of food companies are focusing on sugar reduction or replacing sugar in their products with sugar alternatives and sweet taste enhancers. The latter are molecules that increase the sweet taste sensation but do not taste sweet themselves. Even small amounts of such molecules can potentially replace or reduce the large amounts of table sugar currently added to processed foods, says Coy. Coy and her colleagues are also looking at the other side of the coin, setting up a screening panel of bitter taste tests to search the library for bitterness blockers. These can enhance naturally sweet flavors by blocking the interaction between bitter compounds and human taste receptors. Sensations such as cooling, warming, and tingling are also important to consumers, says Coy. People perceive quality and effectiveness of products from these sensations, so the researchers want to examine sensates, molecules that correspond. These can send important underlying signals to consumers who associate coolness with cleanliness, for example, or hotness with active ingredients. New and more potent ingredients, cooling agents, for example, can be utilized to improve products for the consumer care industry, Coy explains. Researchers at the Biotrans analytical platform are already using mass spectrometry with organoleptics, the study of sensory perception, to look at the chemical compositions that affect the sensory aspects of flavors, fragrances, and metabolites. Coy and the researchers at the Taste Receptor platform want to expand their repertoire of cell system tests to examine sensates for thermoreception and chemical sensitivity. Once useful molecules are found and demonstrated to be safe and scalable, Coy says, then commercial level production can be assessed by colleagues, such as Zhang. Microbe Engine for Industry. While synthesis is still the main source of chemicals for the food and fragrance industries, Zhang firmly believes that in the future, quote, biosynthesis will be on par with chemical synthesis, end quote. Meanwhile, he will continue to focus on three main areas: the discovery of new enzymes and novel metabolites, such as terpenoids, the aromatics used in many scents; enzyme engineering for improved activity, stability, and selectivity; and building on fundamental understandings of biosynthesis. Lindley agrees that there are big changes ahead. He says that even capability building projects such as their work on lycopene always aim for twin outcomes. Quote, to expand our knowledge base and technological toolbox, and also to search for molecules applications that we believe will have industrial potential, end quote. But the biggest challenge has moved quickly to implementation. The conditions used in industrial processes are often far from the evolutionary pressure that has guided microbial evolution over millions of years, says Lindley. The platform will be carefully evaluating, quote, how robust our metabolically engineered microbes will be when we transfer them into industrial scale fermentation strategies, end quote. It means the hard science at Biotrans must be supported by practical industrial considerations, he says, including rational projections and responsive development strategies. The team will also seek to use industrial side streams by converting byproducts and wastes into other value-added products. Biotrans, says Lindley, already has an edge in understanding and being able to exploit areas in which few laboratories have experience. Quote, Biotrans was conceived as an industry-facing initiative able to interact directly and resolve company problem statements efficiently and rapidly, he says. This gives us a clear competitive edge when we are discussing with companies their ambitious plans to put novel molecules on the market, end quote. Singapore, he notes, is also ideally placed, quote, to position itself as the hub for R&D aimed at the Asian consumer and offers the opportunity to develop new molecules that may not be part of plans in the US or Europe. As a result, a new wave of Singapore-made microbe-aided products are likely to hit the shelves in the near future. Quote, we have most of the world's leading food and cosmetic ingredients producers within a few minutes' drive from the lab, and an equally rich diversity of nearby final end-user brand names, notes Lindley. Most, he says, are positioning themselves to service a growing middle class both in Asia and around the world. Populations increasingly able to access information about the impacts of the ingredients they use and willing to invest in healthy, sustainable, and natural products. Now, that article ended with natural products. Does anything about this sound natural to you? I suppose it depends on your definition. Anyway, I recently came across the United Nations Conference on Trade and Development, Synthetic Biology, and its Potential Implications for Biotrade and Access and Benefit Sharing. It spells it right out for you what the United States and Europe, especially, are saying can be considered natural. I'll give you a few examples, but I do hope that you'll go and check it out for yourself. There's a lot of information in there because we're not just talking about food, we're talking about pharmaceuticals and textiles, your clothes, everything, fragrances and cosmetics. They're remaking everything. Here it says, technological developments have subsequently made it possible to mine genetic data from a wide variety of organisms and then to synthesize new genetic constructs that modify the function of living organisms. The growing number of databases containing digital sequence information form an enormous potential catalog of natural parts, functional units of DNA, from which high-value chemical pathways can be discovered or created. Natural parts. This is under implications for biotrade and access and benefit sharing. Chemically synthesized products have been competing with natural products for over a century and a half. For example, the synthesis of indigo by Adolf von Baeyer in 1867 led to the explosive growth of the German and Swiss dye industry, while simultaneously dismantling the import of indigo and other natural dyes from distant tropical locales. However, in some market segments, these products must be labeled as artificial, like artificial flavors or artificial colors in foods. Consumer pressure is leading companies to move away from these products, opening up opportunities for naturally sourced ingredients and biosynthesized analogs. At present, ingredients produced through biotechnological processes can be labeled as natural products, regardless of the production process, so long as the final product does not contain genetically modified organisms. This is under flavors and fragrances. Biotechnology-derived production of flavors and fragrances has expanded rapidly in recent years. The main economic driver behind this trend is the desire to establish reliable and economically profitable production systems that are environmentally benign in comparison with the classic production approaches based on large-scale organic chemical synthesis. A second important driver is that legislation in the European Union and the United States of America allows compounds produced through a living organism to be labeled as natural rather than artificial. I do hope that people will take the time to check out this document because there's so much information in there. I will link to it below, as always, linking to sources below in the notes. But there's so much in here on patchouli and stevia and orange and there's so much more than I can say. So there's just so much in there that you really should check it out. There's another section under pharma and phytopharmaceuticals where they're talking about artemisinin, and that this is a plant, or it's derived from a plant, that's been used in Chinese medicine for at least 2,000 years and is being used around the globe to treat malaria. And they say, to address price and supply fluctuations resulting from natural production, significant work has been carried out since 2004 on synthetic biology production techniques. In 2005, the Bill and Melinda Gates Foundation granted funds to the Institute for One World Health to finance the development by Amyris of genetically engineered yeast that could make artemisinic acid, a precursor compound that can be chemically synthesized into artemisinin. Here's one table that has examples of synthetic biology, synbio products that could replace naturally sourced biological resources. I've seen quite a few charts like this, but yeah, look there, there's that resveratrol. That was pretty popular. Yeah, that's already been commercialized. And then we come to food labeling. In the United States and Europe, synbio products can be labeled as natural ingredients, but this is not necessarily consistent with consumer perceptions. As demonstrated in a survey conducted by Consumer Reports in 2014, consumers widely misunderstood the term natural, and about two-thirds of consumers believe the term means a processed food has no artificial ingredients, pesticides, or genetically modified organisms, and more than 80% believe that it should mean those things. Then it goes on to speak of the United States. In 2015, the FDA sought public comment on the definition of the term natural for human food products, including foods that are genetically engineered or contain ingredients produced through the use of genetic engineering. This was in response to several citizen petitions, as well as 3 federal district courts that referred to us for an administrative determination under 21 CFR 10.25c. The question of whether food products containing ingredients produced using bioengineering may be labeled as natural, all natural, and or 100% natural. The public comment period closed on May 10, 2016, but the FDA's next steps are unclear. Thus far, the FDA has not made a statement about this subject, an adjustment to its policy, or an administrative determination, but rather restated that in accordance with the original guidance, it does not object to the use of the term natural on products that do not contain added color, artificial flavors, or synthetic substances. In late 2018, the United States Department of Agriculture published its final rule implementing the National Bioengineered Food Disclosure Standard signed into law in 2016. And I'm skipping ahead here. The final rule defines bioengineered food as any food that, quote, contains genetic material that has been modified through in vitro recombinant DNA techniques and for which the modification could not otherwise be obtained through conventional breeding or found in nature, and excludes any genetically modified material that is not detectable. Non-detectable amounts of modified genetic material do not require labeling. End quote. Since synthetic biology products do not fall within the definition of bioengineered food or the list of bioengineered food products, they are not affected by the labeling requirements in the National Bioengineered Food Disclosure Standard. In the European Union, foods must state on the label if they contain or consist of GMOs or contain ingredients produced from GMOs. However, foods produced with the help of genetically modified microorganisms, for example, synthetic biology products, do not have to be labeled. The natural label for flavoring substances is regulated more strictly in the European Union than in the United States. Flavoring substances can only be termed natural if they are, quote, obtained by appropriate physical, enzymatic, or microbial processes from material of vegetable, animal, or microbiological origin, either in the raw state or after processing for human consumption by one or more of the traditional food preparation processes. Natural flavoring substances correspond to substances that are naturally present and have been identified in nature. Synthetic biology ingredients correspond both to substances that are naturally present and have been identified in nature and are obtained by microbiological processes from material of vegetable origin. This would suggest that they could thus be labeled as a natural flavoring substance under this regulation. And then, under general conclusions on possible implications of synthetic biology for biotrade, says, furthermore, the main feedstocks used today for synbio require extensive agriculture, which is now the leading threat to global biodiversity. As synbio becomes increasingly accessible, it is possible that practices could shift from large-scale cultivation of crops used for a single fragrance or flavoring to the cultivation of crops such as sugarcane or any other crop that can be efficiently processed into a feedstock to supply carbon to microbial fermentations. Farmland in certain developing countries has already been converted to sugarcane production to meet the needs of synbio companies. This conversion can impact food prices and availability for the most vulnerable populations. Furthermore, some synbio applications are aiming to convert low-value forests and agricultural products into feedstocks, which would also have harmful effects on biodiversity. If synbio products are to have their promised benefits, they must have low environmental impacts, use biological feedstocks sustainably, and operate according to high safety standards with respect to humans, animals, and the environment. And we already know that there are companies that are already using carbon capture, carbon dioxide from the atmosphere or from smokestacks to feed that to their microbes because it's more sustainable. Here's a good part. Commercial actors recognize that consumer preference for natural ingredients has become a key driver in the marketplace. The commitment of numerous major multinational corporations to transition to sustainable sourcing, natural ingredients, and clean labels could be a boon for biotrade if the natural ingredients are naturally procured from sustainable sources. Alternatively, if regulations continue to consider synbio products as natural, this transition may have no or harmful effects due to a product substitution or displacement. Some enterprises will decide to choose to use high-cost naturally sourced products like vanilla bean extract due to their higher value in the marketplace, while others will use lower-cost synbio products that allow products to be labeled natural without a commitment to a sustainable biodiversity-based sourcing model. The question for companies will be whether there is a comparative quality advantage that justifies the higher price for a naturally sourced product. This likely will be a sector-specific decision. If a cost-benefit analysis demonstrates that the natural synbio product is satisfactory for the manufacturer's purposes, the naturally sourced product will not be the preferred option. Under potential implications of synbio for access and benefit sharing, it says academics and companies can now easily take genetic code that has been uploaded to the internet and using a DNA synthesizer recreate and modify that code to produce new substances, tests, and perhaps even new organisms with no meaningful way to track the origin of the genetic information that formed the basis for the discovery. In case I haven't covered it yet, there's many definitions for synthetic biology, but this one is found in the 2017 report of the United States Committee on Future Biotechnology Products. It defines synthetic biology as the application of engineering principles to reduce genetics into DNA parts so that those parts can be understood in terms of how they can be combined to build desired functions in living cells. I mean, really, people, you should take a look at this document because it goes into the biobricks, which they talk about a lot in George Church and Ed Regis's Regenesis. And yeah, it's just pieces and parts that they can put together because they've been engineered to click together like Legos. Maybe you'd like to read it. So I'll link to it below. And I know this was a longer video, but when I got to the end of this great article and it's talking about how these ingredients are for healthy and sustainable and natural products, I just had to tell you about the United Nations document here, the United Nations Conference on Trade and Development, and just what the United States and just what Europe are saying. What's natural? All this stuff. All this stuff. It's more important than ever that you find local farmers and people that you can trust are actually doing farming. And I don't mean this high tech, the new IoT farming, like these millennial farmers and these huge YouTubers that are showing you have to have all these big pieces of connected equipment and technology. No, people that are working with the planet and working with the life that grows here. And it really, to me, it seems like we are we're on the cusp of this new world, and that there's two roads, they're diverging, and we need to actively choose the path of nature, or we should just buckle in for what they're about to do to us. So please find a farmer. Eat Wild, that's one website. I'll link it below. Real Milk, that's another. I know not everybody drinks milk. Let's everybody get along. But it's just everything is under this, and it's so big. It's so big. So please take a look at my other work. Tell your friends. Please tell people that you interact with. Tell YouTube. tell the people that are talking about these new foods, these foods that they're able to make taste like, look like, smell like, feel like the real thing, but they're not the real thing, and all the other science experiments that they're trying to feed you. So, thank you for listening if you got this far, and I really hope that this helped you to see that I'm not just talking nonsense over here. This is the UN. This is scientists talking to you about this. So
they use water and carbon dioxide to feed microbes that output a single cell protein that has the same nutritional content as soybean. So that means you can basically replace soybean production, which uses an astronomical amount of our land today, with something that is produced in a fermentation cylinder and just uses water and carbon dioxide.
Please tell people.